This is a video looking at the topic B6 and some triple content, protein synthesis. So, we have been talking for such a long time now about protein synthesis occurring at the ribosomes. Now we're actually going to talk about what protein synthesis is. So on the left-hand side of this diagram, we've got the nucleus. So we're deep inside the cell at the moment. We've got the nucleus on the left, and on the right bottom corner, we've got a ribosome. Now, what happens in the nucleus is the DNA temporarily unzips. So those hydrogen bonds holding the base pairs together temporarily unzip, which causes no change to the DNA at all. We need to go back to B2. We eat protein. The protease enzymes, for example, pepsin and trypsin, break down that protein to make it small and soluble. If it's small and soluble, it can be absorbed into the blood, and then those amino acids move around in the blood until they get to cells where protein synthesis can occur. And then protein synthesis is about taking those amino acids and building them back up into proteins. Okay? And really, the only reason we've broken them down is to get them through that wall of the small intestine, through the villi, into the blood, and also to build different proteins. So, the DNA in the nucleus has temporarily unzipped. Amino acids are attracted to that unzipped DNA by those hydrogen bonds, and they match up A to T, C to G. So we can see now we've got a template. Okay, the template is just here. And that template will then pull away from the DNA, so the hydrogen bonds will weaken, so the template pulls away. The DNA zips back together in the nucleus, and that template then makes its way over to the ribosomes. So here is the template, and here it's made its way over to the ribosomes. When it gets to the ribosomes, it's still only half the protein. It hasn't had those base pairs join up yet, so they're just single bases lined up waiting for their corresponding pair. So what happens here is carrier molecules, these, bring over the corresponding amino acids. They join up to the template with those hydrogen bonds, and that makes the protein. And the protein will coil up, and examples of proteins are hormones, enzymes, proteins that are used for growth and repair, any kind of protein you can think of. So here we have it written as a summary. So we'll go through it again with the words here. So the DNA in the nucleus unzips. This exposes those bases. The amino acids drift over and are attracted by those hydrogen bonds to the DNA. Once the template has been made, it pulls away from that DNA and the DNA zips up again. The template then drifts over to the ribosomes. Ribosomes is where protein synthesis happens. Think rack of ribs made of protein, so ribosomes, protein synthesis. And the corresponding bases as amino acids are brought over by the carrier molecules, and they match up to the template. And this creates the protein. Okay, and those examples of those proteins could be enzymes, hormones, or other protein structures. Hope you found that useful. Thank you.